Welcome back to Physiology. This third lecture covers chapters 3 and 4, cell structure and enzymes. Now, a lot of this chapter might be review, and so I'll focus on some of the general aspects today and talk about some things that you should review on your own at home. First, I'll start off with today's study tip. After reading a page in the book, close the book. Then, from memory, try to write down a few sentences and key words about the reading. You can see what you remember and see what you understand. Very often as we're reading, you might read the book, but then it's not actually sinking in and you're not getting the information. So try this as you're studying through these chapters and see what you need to focus on. Okay, so getting started with chapter 3, inside the cell. Inside our cells, we have a liquid component, the cytosol, or called the cytoplasm. And this is gelatinous aqueous mass inside the cell. And it's not just water. Right? It has suspended proteins and organelles, the little organ-like structures inside the cell that have a specific function. It also acts as a matrix for chemical reactions to occur. So we're going to talk a lot about these chemical reactions this semester. We're going to talk about metabolism, cell metabolism, and some of the reactions occurring inside cells. Some cells contain fat or glycogen, storing energy. Inside the cell is also a cytoskeleton. So this is the structural component of the cell that helps create the shape of each individual kind of cell. There are proteins that help keep the organelles in place and also give structure to the cells. Now here's a view of the inside of the cell. The cytoskeleton you can see as these crossbars and filaments all throughout the cytoplasm. And we'll talk about the three different components of the cytoskeleton, but here you can see these microtubules which provide this structural support for the cell. Also in this picture you can see the outside, that plasma membrane. Remember, it's made of phospholipids, a phospholipid bilayer. And here we start to see some of the organelles present. Mitochondria, ER, the nucleus, which is the nuclear envelope on the outside, and ribosomes, these little pink dots that are studded on the ER. So we said the cytoskeleton is made up of a couple different kinds of filaments. There are microtubules. And here is a fluorescent microscopic image of the microtubules labeled here in green. Okay. These are the biggest of our cytoskeletal elements. They give structure to things like cilia and flagella. So you can think about single cells that have cilia or flagella to move, or ciliated cells in our bodies, right? Things like the surface lining of the lungs have ciliated cells to beat and push that mucus back out of the lungs. Microfilaments, however, are a little bit smaller than microtubules, and they also give structure, but they're giving structure specifically to things like microvilli in the small intestine. So both structural components, different sizes, and have slightly different functions in terms of what they can give structure to. You might have heard of some of these components. Actin and myosin are muscle filaments that we'll talk a lot more about in a later chapter. Those are microfilaments. Okay. And so you at home are going to need to go over these organelles. I'm not going to go over all of them in detail right now because this is probably review, and if it's not review or you do need to review, you'll have some time to do so at home. So make sure you know the identity, what these things look like, and the function of each one. Okay, so I can ask you about what the ER is doing, what the mitochondria are doing, what the Golgi complex is doing. You should know generally what the organelle's function is. And then as we talk about these different organelles and their role, I'm just going to assume that you know what these organelles are doing inside the cell. Okay. Take a little closer look at the plasma membrane. I put a YouTube link up here too that you can watch at home. Okay. Remember, these slides are on Canvas, so you can access them there and access these links there as well. And we said that our plasma membranes are made of phospholipids. So here's the phospholipid bilayer, right? There's two layers of phospholipids. And you'll note that these polar heads are facing the extracellular surface and the intracellular surface. So the surface on the inside of the cell and the surface on the outside of the cell are both made of these polar heads of phospholipids. The tails then that are hydrophobic, those all line up in the middle. Okay? And sort of between and among these tails are cholesterol components. protein components, these are going to be things like receptors or channels and transporters that actually are moving through the phospholipids. So they're embedded and they have a side that's contacting the intracellular side and extracellular side of this protein. Those proteins can be attached to glycoproteins, glycolipids on the surface of the cell. And see, the surface is not smooth, right? The surface of the cell has these proteins, glycoproteins, glycolipids, carbohydrates on here too. And this all makes this extracellular matrix, we call it. Okay, so lots of different components of a plasma membrane. And we call this a fluid mosaic model, one, because it has a lot of different components, all these things in the cell membrane. And the fluid part of this is that these things are not just stuck there. So your cells can insert more receptors, take them out, open and close channels, move things around. And the phospholipids themselves actually are moving and changing places throughout this membrane too. So it is a fluid mosaic model. Okay. So our basic structure, phospholipids are really important. We just said this in the other picture, right? Head is hydrophilic, faces outward. Hydrophilic means it loves water. The fatty acid tails that are hydrophobic then face inward and they fear water. And so we end up with that double layer. The cholesterol that is in the membrane as well is mostly hydrophobic, and so it hangs out among those hydrophobic tails of the phospholipids. And it helps to stabilize the membrane. So it's an important component of our cell membrane. It helps stabilize the membrane. The proteins that are there, we said, have different components, and they're embedded throughout those phospholipids. The hydrophobic amino acids stay inside the membrane, whereas hydrophilic pieces of the amino acids can be in the inside and outside of that membrane. And those carbohydrates and things that we saw on the surface of the cells help for cell recognition. They're cell surface markers. And so you can think about something like a bunch of liver cells are going to have to communicate with each other and work together to be a liver tissue. And so the carbohydrates on the surface are part of that cell recognition function. Okay, so that was our very brief foray into cell structure. There's lots of YouTube videos online as well. You can find lots of good review, but make sure you review what cell structure looks like and those basic components and organelles. Now we're going to get into another review topic, and this is going to be protein synthesis. So again, in a past class you've had this, maybe your intro bio class, maybe a different class you've taken. So this should be some review, but we'll talk about it again here briefly. And protein synthesis is really important. Our body has to make proteins. It has to make lots of different kinds of proteins. So to remember how cells do that, we think about the whole cell, right? So this big square here is a cell, and here's the nuclear envelope. Remember, the nuclear envelope contains your DNA, and the DNA is the genetic code. So it stays in the nucleus and is the code for you and all the proteins that make up you. What then happens is that DNA, when your body wants to make a protein, that DNA is transcribed, meaning that the cell reads it and makes an mRNA molecule that corresponds to that specific gene or genes in the DNA. The mRNA then is what exits the nucleus, finds a ribosome, and is translated, meaning that the information encoded in the mRNA is read to make a protein. So remember last time, chapter 2, we talked about making amino acid chains that then fold into proteins. And so this process is, we sometimes call it the central dogma of cell biology because it's transcription and translation. The information is carried from the DNA into mRNA and then into protein. I want to point out here that the DNA is not turning into mRNA and that's not turning into a protein. The information is moving through those three steps. So the DNA always exists, stays in the nucleus. The DNA is read by cellular machinery, and an mRNA molecule is transcribed or created based on that information. DNA stays here, mRNA leaves. mRNA is translated, which means your ribosome reads that mRNA and creates a protein based on the mRNA. but it's not turning DNA into mRNA or into protein. Okay, the information is being brought through those 3 steps. Okay, so a little bit more depth about how this works. Here's our DNA molecule. And remember that DNA has those base pairs in the middle, right? So here's A pairing with T, C pairing with G, and this double helix has that sugar phosphate backbone wrapped around. Okay. In transcription, what happens is the cellular machinery comes in and it adds on nucleotides. It adds on base pairs to this open strand of DNA. So the DNA strand is opened and base pairs are added on. Remember that RNA has U instead of T, so uracil instead of thymine. And what that means then is that when the machinery reads an A on the DNA, it doesn't put a T there, it puts a U. But just like DNA, when it sees a C, it adds a G. When it sees an A, it adds a U. And now this growing RNA strand then corresponds to this DNA strand, right? It was made off base pairing rules. And what you'll see is that this RNA strand then matches this DNA strand, except for it has U instead of T. Okay. So this is taking place in the nucleus. This whole process is called transcription. RNA polymerase is an enzyme that binds to what we call a promoter region. So at the beginning of a gene, if you want to transcribe a gene like hemoglobin, maybe you want to make more hemoglobin for your red blood cells, there's a promoter region at the beginning of that gene. RNA polymerase goes to that promoter region, binds to the promoter region, and then as the DNA unwinds, RNA polymerase goes through and adds on those base pairs corresponding to the ones in the DNA molecule. Okay? And so I'm already seeing a typo here in my PowerPoint, right? Because this says that RNA makes complementary base pairs. If template has an A, RNA will get a T. Well, in theory it would, except that RNA gets U instead of T. So this should say if template has A, RNA will get a U. What happens is that RNA polymerase adds on base pairs until it gets to what's called the terminator region. So those promoter and terminator regions are sandwiching your gene of interest. And then that whole strand of DNA, the corresponding mRNA strand is made. Okay. Once that mRNA strand is produced, there's some other changes that take place. So there's things added and taken out and spliced from that mRNA. So the mature mRNA transcript is what actually leaves the nucleus and goes into the cytoplasm to find a ribosome. Okay. So I put another video animation on here. It's only about 7 minutes long, but it's going to help you review translation and transcription. And now I want to talk briefly about this translation point. So now we've made the mRNA from a gene, and we're going to take it, leave this nucleus, and it's going to be made, a protein is going to be made based on that mRNA molecule. Okay? So when you want to translate, when your cells, your ribosomes, want to read that mRNA and make a protein, what happens is that the ribosomes have to come together around that mRNA molecule. Okay. Then to start it, you need to have tRNA. So the second kind of RNA, tRNA or transfer RNA, is going to be the RNA that reads the mRNA molecule and starts to add amino acids onto that chain. Okay. And this is, translation occurs when your ribosome and the tRNA reads 3 nucleotide bits called codons. So it doesn't say, oh, there's an A here, I'm going to put an amino acid. It's a U here, I'm going to put a second amino acid. It says, okay, right now I see AUG. I'm going to put on one amino acid that corresponds to AUG. Oh, now I have UUC. Okay, well, what amino acid corresponds to UUC? And so it reads in these 3 nucleotide bits called codons that are read by that tRNA. And then it adds the amino acids all in a chain. Okay. Then we said that amino acids are put together through peptide bonds. So the peptide bonds form between the amino acids while they are still attached to the ribosome. And that's creating your amino acid chain that will become a protein. Okay. Then the mRNA moves, and the next codon is on the ribosome. So I think it's easier to see a picture of it. Okay. Here you've got the mRNA strand here in pink. Our tRNAs are green. What we see is that each tRNA has 3 little prongs. So if the codons are the 3 letters on the mRNA, on the tRNA, the 3 letters here are called an anticodon. Okay? So a tRNA comes along and base pairs with this codon, and it's a coding system that adds this amino acid to this polypeptide chain. Okay? And so that continues along. This codon is red. As it moves over, then you're going to have this next 3 nucleotide codon red, and different tRNA comes along and adds the amino acid to the chain. And it adds and adds and adds as long as this chain is, right? And then eventually, in the mRNA, there'll be something called a stop codon. So it's a 3 nucleotide codon that not a tRNA binds to, but something called a release factor. So when the release factor is bound here onto one of the codons on the mRNA, all the stuff pops off. The ribosome, not degrades, but the ribosome breaks apart, and now you have a free polypeptide chain, which will then fold and do all the folding we talked about to become a protein. Okay? So here's everything all together. You've got your DNA in the nucleus, mRNA being produced in transcription by base pairing with the DNA. The mature mRNA molecule then leaves the nucleus, finds a ribosome. tRNAs pair their anticodon with a codon on the mRNA to add on a specific amino acid to a growing chain. When that stop codon is reached, this all falls apart. Now you have an amino acid chain, which is the primary structure of your protein, and the protein folds to become a mature protein. Okay, so that's our very brief review of Chapter 3. There's more stuff in there. They packed everything into Chapter 3. You also have meiosis and mitosis. So this is cell division, and I want you to review this on your own at home, including watching this video. Okay, so again, lots of great resources for this stuff online. You can read about cell structure, mitosis, meiosis, but I tried to give you some videos that I watched that are pretty good about these topics. I also want you to read more in detail about RNA and DNA. And so review all this stuff that you learned in general biology. Okay. I also want you to think about and review DNA replication, which has to happen before meiosis and mitosis, right? So there's a whole process of replicating DNA synthesis that occurs before the cells are going to divide. So review that as well. Okay, lots of review this week. Okay, so take a break if you need to, then come on back for Chapter 4. All right, Chapter 4, enzymes. Enzymes are proteins. Specifically, they're proteins that catalyze a reaction. And so we start thinking about these chemical reactions happening in our bodies. They all could occur without the help of enzymes, but they would be really, really, really slow. And our bodies don't want to be slow. They want to be fast. They want things to move faster. So we have to have enzymes that catalyze or speed up our reactions. They don't cause the reaction, but they speed it up so we can do things faster. They increase the rate of reaction. And they do so, we'll talk about how they do that a little bit, okay? Just like for any other protein, form determines function, okay? So their shape determines their function, and therefore they're highly specific for certain substrates. The substrate is the thing that's going to bind to the enzyme. It's this lock and key kind of model. So this enzyme here is only going to bind this kind of substrate and catalyze this specific reaction. In this case, is breaking it apart. So see this, the substrate here, the reaction is the substrate being broken up into 2 pieces, which is accomplished with the help of this enzyme. Okay. So here is an example with our enzyme here, okay? And let's say we have two substrates, substrate A, substrate B. Those are going to bind specifically to this active site in the enzyme, forming what we call an enzyme-substrate complex. So here's the enzyme and the substrate. It's a complex. They're bound together. And because the shape of this enzyme is forcing substrate A and B to interact, reactions happen here at the interface between A and B. And the products of this reaction then are a smaller product C and a larger product D. Okay. What's important to note here is that the enzyme isn't changed, right? So A and B bind, stuff happens, C and D are produced, and then they pop off and go do whatever they're going to do. The enzyme doesn't change. That means that the next A and B that come along, same thing. So enzymes, being stable proteins that don't change, can catalyze reactions over and over and over again. They can be reused. Okay, quick quiz. Try not to look at the next slide. Let's think about some of the mechanisms of enzymatic action. Okay, so take a second, see if you can fill in some of these blanks here. Okay, so substrate binds to the active site of the enzyme. The enzyme-substrate complex is formed. There's a change in arrangement of the bonds of the substrate. So something happens, some kind of chemical reaction, change in bonds happens at that the substrate interface. Okay, the product is released from the enzyme, and our enzyme is free, meaning it's not used up. Okay, so why are enzymes catalysts? We said that they can make reactions happen faster, but they don't cause reactions. So how we do this is based on something called collision theory. Okay, so chemical reactions can occur when atoms or molecules collide. And so if you think back to our chemistry or basic biology, remember that chemicals, molecules, atoms are always moving. Okay, so all the molecules in your body, surfaces, and objects are just like moving around. Okay, and we have to think about energy, and we have to think about orientation. Okay, so there's a certain amount of what's called activation energy needed to disrupt or rearrange stable electronic configurations. Okay, and so the reaction rate is the frequency of collision, how fast these things hit each other, with enough energy to bring about a reaction. Okay, and so this starts to sound complicated in like physics, and it is, but we can think about reactions happen faster when you have increases in temperature, increases in pressure, increases in enzyme concentration, or increases in reactant concentration. And these little bars here, that is indicative of concentration. Okay, so things are bouncing around. If they hit each other more, they're more likely to interact and cause that product to occur. Okay, and so why temperature speeds up reactions is that the molecules are moving faster. That's what heat is. Molecules are moving faster, and as they hit each other more often, you're more likely to get the products. Enzymes speed up reaction rates, production of products, by affecting this activation energy, the energy required here. Okay, so here's what it looks like in a graph. We've got our energy level here on this y-axis, okay, and this is going to be time on our x-axis. And so here is our substrates, okay, so you had substrate A, substrate B. They're called substrates or reactants, either way. And if we don't have an enzyme, then it takes a lot of energy to initiate this reaction, to go from our reactants A and B to our products over here. Okay? Our enzymes reduce activation energy. By reducing activation energy, that reaction then can happen faster. Okay, so again, this reaction would happen without an enzyme. It just might take a really long time. With an enzyme, activation energy is reduced, and now it's more likely for this reaction to occur. So look at this graph, stare at it for a while, think about it. Okay. This topic I probably wouldn't look up on YouTube because you're going to quickly get into very, very complicated chemical and physical dynamics of enzymes. For our purposes in biology, we care about this, that reactions are going to happen. Reactions with enzymes happen slower or are less likely to happen than those with enzymes because enzymes lower the activation energy. Okay? So, it gets more complicated. We've got lots of enzymes. We also have lots of coenzymes and cofactors. These are going to be things that are required for enzymes to work. Okay? So cofactors are things like metal ions, like magnesium. Coenzymes derived from vitamins, where you have to have an enzyme to make your reaction go faster, but you also have to have a coenzyme or cofactor. Okay? And so some of these things, like here's something called an apoenzyme. This protein portion is inactive until it's bound by its cofactor here. And then our substrate can bind to the active site here and allow that reaction to occur. Okay. So enzyme names usually end in ASE or ACE. So you can hear about helicase, polymerase. These are all ACEs. So you know ASE is going to be an enzyme. Lactate dehydrogenase, another example. This is one that removes hydrogens from lactic acid. And we can diagnose some diseases based on enzymes present in the blood. Okay, things like creatine phosphokinase, CPK. Okay, so we'll learn about some of these kind of clinical applications of this enzyme measurement too. But probably this will mostly come up when you start working in clinics and see for your specific area of expertise what that might look like. Okay. For enzymes to function, they have to have an optimal pH and temperature. And this doesn't mean that they can't work at others, but it means that we have an optimal pH and temperature for each enzyme. Okay, they work fast under those conditions, and we measure fast by looking at the reaction rate. Okay? All right. So enzymatic activity as measured by reaction rate, how fast you're producing a product of that reaction. And here's a typical temperature curve for an enzyme, okay? So meaning that this enzyme is most active around 37 degrees Celsius. And what you see here is two things. One is that the increase in activity here is kind of a slow increase based on temperature until you get to that peak. Why? Because as you increase temperature, the molecules move around more and they're more likely to interact with the active site. Okay? But you quickly see here a drop-off in activity past the peak. That's because at high temperatures, proteins, like enzymes, denature. Right? And remember, when something denatures, the protein falls apart, loses its structure, and we have a loss of structure, we have a loss of function. So this is showing denaturation of a protein by high temperatures, and so our enzyme can't act anymore. Okay. So similarly, we have something happen with pH, although pH looks more like an even bell curve on both sides, right? The temperature one, we see this skewed curve where it quickly drops off past that, past the peak for heat. pH, we have an optimal pH of 5 in this case, but the enzyme does work in a wider range of pHs typically, and then drops off quickly on both sides. Okay. Another factor besides temperature and pH that affects enzyme activity is going to be substrate concentration. And this kind of makes sense, right? You put more stuff into the reaction, more stuff comes out. Okay? However, it's only to a point. Okay? So we increase substrate, you have more enzyme activity, but then it levels off. Why? Because at some point, all of the active sites of the enzyme that you have present in the cell or in the body are going to be occupied. And when that occurs, you can throw all the substrate you want at those enzymes, but they can't hold anymore. So they can only move so fast. Okay. So this chapter, and we guys are thinking about something called bioenergetics, the flow of energy in living systems. Okay. And we said we had to think about energy, right? So you have to think about endergonic reactions, reactions that require energy input, and exergonic reactions, those that release energy. And some of you are saying, "Oh, this is physics." Yeah, it's physics. We're based in physics, right? We have to abide by the rules of physics in biology. Okay, and so we've got reactions that require energy and reactions that release energy. Okay, remember that cellular energy is ATP, made by ATP synthase. Synthase, that's an enzyme. You can tell because it's ASE here. Okay, and what ATP synthase does is it takes the substrate, ADP and P, this inorganic phosphate, and it makes ATP. So this enzyme is critically important making ATP. All right. And so we start talking about the chemical reaction part of this and talking about oxidation reduction reactions. Okay. And so these are paired reactions that are necessary to do things like make ATP. So we're not going to go into detail with cell respiration this semester, but remember back to your previous biology courses that ATP is made when you take glucose, put it in this whole series of reactions, that is cell respiration in a mitochondria, right? And eventually producing ATP. So all along those stages of cell respiration, you have these oxidation reduction reactions, okay? We're moving around electrons. And so when we move these things around, to gain is to be reduced, to donate is to be oxidized, okay? So I have my students, some people like to remember it as oil rig, oxidation is losing, reduction is gaining. And so these are paired, right? Because when an electron moves and it's lost from something that's oxidized, it's gained by something else. And that's what's being reduced. Okay? So here's an example. Here's an example of something that happens in the Krebs cycle in cell respiration. You have succinic acid turned into fumaric acid. Okay? And what this does is that we go from FAD to FADH2. Okay? All right, let's think about this. What's oxidized here? We said we're moving stuff around, right? We're moving around these electrons. Okay, so here we go. We are moving something from FAD and taking FAD and making FADH2. We got to move the electrons from here, succinic acid, to the FADH2. Which means that we got three things to think about. Something's being oxidized, something's being reduced, and when it's happening, there are atoms removed. Okay. So, succinic acid is the thing that's being oxidized because it's losing. Oxidation is losing. Okay? What's reduced then is the thing that's gaining because reduction is gaining. So what's gaining? FADH2. And the atoms that are removed are these H+, these hydrogen ions. Okay? Okay. So we can think about these kinds of reactions. We have oxidation and reduction reactions, and they're happening throughout the body, throughout cells. Okay? And so think about all those things in this chapter. Certainly come see me in office hours if you have questions. We can talk a lot more about these topics. All right, see you soon.